Are you taking AP Biology? I'm not, but if you are, you're in the right place because today we're going to be going over Unit 1, the chemistry of life. We are joined by Miss Tiffany Jones from AP Bio Penguins. How are you doing today, Tiffany?
I am wonderful. Just trying to get the YouTube pulled up so that I can see the chat for when we do get started.
Amazing.
But we are
And
started.
great. And as you are all coming into this chat section, if you're watching a recording of it or you're here live with us, talk to us, ask us questions. We're here to answer them. At Marco Learning, we support AP students with tons of resources, but one of the best things we have ever found on Instagram and on TikTok and on the interwebs is AP Bio Penguins. So Tiffany, I'm going to turn it over to you. We're going to walk through Unit 1. A lot of people have forgotten the content from Unit 1, but this is going to be a great review. So I'll let you share your screen. And again, I encourage you, if you are watching along, chat with us in the chat in the comments and questions. And if you like this video, go ahead and smash that like button, subscribe to our channel. We're going to be going live all through the spring.
Well, I think yes. Hi, welcome. So I always started out with what's up, AP Bio Penguins. So today we're going to be looking at Unit 1, which is all about the chemistry of life. And so I do have my handle on there on AP Bio Penguins. I have that same handle on Instagram. I have it on TikTok. I have it on Twitter. Basically, if you go to Google and type AP Bio Penguins, you're going to find me. And so why are we penguins? Like, why do I have this whole idea of penguins? Well, because penguins are dressed for success. And you, as an AP student, you're dressed for success and you are going to rock this AP exam. You are ready, you're prepared, and you're going to be amazing. So I always like to tell y'all that you are an AP Bio Penguin now. So today's plan. Now, I'm kind of switching things up a little bit because of the fact that I already do unit reviews. They're all found on my website. They're all found on my YouTube channel. So I did want to switch it up. And the original weekend review that I did for my students kind of back in the day, I used to go to Panera. And so I used to call these my Panera sessions. And at these sessions, I would take a piece of paper and I would just graphically draw everything out. They would throw information at me and we would just make a graphic organizer. So my thought was, let me just do that live with y'all. I would make a graphic organizer and it can help us to walk through all of the information. So my chat is open. So if you come across a question, you're more than welcome to throw it into the chat and I can answer as we go. But we're going to start with macromolecules. We'll then talk about water properties. I've got a couple practice questions, and then we have the Q&A, or the time that you can ask questions, and I can try to help you with whatever your questions are. And so this could be a little weird as I try to work through this new system, but I think it will be okay. So macromolecules, there are 4 different macromolecules that we have. You have carbohydrates, you have proteins, you have nucleic acids, and you have lipids. Okay, and it's important to know these 4 macromolecules. And when you're starting with Unit 1, they seem a little complex with all these moving parts, right? And so as you work through the year, and so now that y'all are in February, depending on when you're watching this, maybe May 10th, the night before the exam, you have a better grasp of all of this stuff. So you can apply it in the later units. So let's start with carbohydrates. So carbohydrates are made up of carbon, hydrogen, and oxygen. So it's important to know the elements that are in each of these macromolecules. And they have a 1 to 2 to 1 ratio. Think about glucose, C6H12O6. It's a 1 to 2 to 1 ratio. And it's because each of those carbons is bound to 2 hydrogens and then also the oxygen. And so their monomer, because it's important to know the monomers, is a monosaccharide. So an example of one of those is, of course, glucose. So you can kind of see this is the general structure that you'll see for a carbohydrate. It's going to have that ring structure. There'll be an oxygen here, and then, of course, the carbons making up that ring. And so some other examples, you have fructose, you have galactose. Now, we've talked before about that there's this thing called a dehydration reaction. And dehydration reaction is when you're going to remove water to form a bond. So what is it called if I have more than one monomer together, or more than one of these monosaccharides? Well, it's going to make a disaccharide. So it's 2 of those monosaccharides. And some examples of those would be, of course, your sucrose, your lactose, and your maltose. Sucrose is made up of glucose and fructose. Lactose is made up of glucose and galactose. Maltose is made up of 2 glucose molecules. Now, it's not directly in the standards, but you may see this, a lot of vocabulary when you're reading through the questions. And one of those words you might see is a glycosidic linkage. That is just the bond you'll see between these monomers, between these monosaccharides. It's just called the glycosidic linkage. And so there are also polysaccharides, which are just long strings of these monosaccharides. And so they have two different kind of functions to them. They can either have a structural function in which like cellulose is part of the cell wall in a plant and kind of gives that structure and rigidity to the plant. Or it can be chitin. Chitin is found in arthropods. So think about like stepping on a cockroach and that crunch noise. That's due to the structure of chitin that's in that shell, the exoskeleton. And it's also in our fungi cell walls. We also have storage polysaccharides. These are going to be to store that glucose because, as we all know, glucose is needed for cellular respiration. And so if you don't need that glucose right now, you might want to store it for later. And so plants will store theirs in the form of starch. Think about a potato. Like someone always tells you that potatoes are very starchy, right? And then animals are going to use glycogen. Glycogen is going to, of course, be a string of glucose, and it's stored in our liver. And so we're going to use a hormone called glucagon when we're trying to break down the glycogen. So there's a lot of these words that all sound the same. And so it's important to know, like, in terms of the bonding, because we know that starch and cellulose are both in plants, but we can break down starch, but we can't break down cellulose. And so there's a structural reason for this. And when I say we, I mean like animals. So if you look at the structure for starch, it is a 1,4 alpha linkage. So that means that when you look at the starch, see, here's our oxygen. It's on the same orientation. And so all of our fifth carbons are kind of up. Sorry, not fifth carbon, sixth carbon are all in that upward formation. But when we look at cellulose, now that oxygen kind of switches. And so you see like this back and forth that we have with that, okay? Because it has a 1,4 beta linkage. And so we as animals can break the 1,4 alpha, but we can't break the 1,4 beta. And so it just has to do with a structural component. So that gives us our gist of carbohydrates. So now let's look at proteins. Okay, now we've thought proteins are huge. You talk about those all in gene expression. You talk about those in evolution. You talk about proteins all year long, right? So what are they made of? Carbon, oxygen, and hydrogen are going to be the commonalities or in everything, right? You have nitrogen, and then you sometimes have sulfur. It's in a lot more than just sometimes, so we just say sulfur. And so the nitrogen is going to be found in this amino group, and then the sulfur will be found in this R group. And there was a big important experiment back in the day, a Hershey and Chase experiment, in which they were trying to figure out whether the material, that transformed material, like so the hereditary material, whether it was basically whether it was proteins or whether it was nucleic acids. And so they were able to determine that since sulfur is in a protein, that it was not the transforming agent. And we'll talk in a second about the other side of that. And so the monomer for it is an amino acid. Amino acids are made up of basically 4 different things. You have this central carbon here, and then you have this amine group. So the NH2 is your amino group. It's very basic. Okay, and so it's going to cause the hydrogens to be removed from the solution to allow for that basic component to it. It also has a hydrogen. And then there's a carboxyl group. As you notice here, we have a C double bond O, OH, which is the carboxyl group, and it is very acidic. Okay, so between these two, the amine group being basic and the carboxyl group being acidic, it has a lot of variance based on pH. And then we have this variable group, this R group. This R group can be one of 20 different things. And I put 20 in quotes because of the fact that there's actually more than 20 amino acids, but we really just kind of focus on those 20. So the R group is variable. It can differ, it can vary based on each amino acid. And so there's a lot of different things we'll talk about in just a moment about that R group. So when we go through the dehydration reaction and we take out that water molecule and we put together 2 amino acids, that's called a peptide bond. Okay, so you're going to have multiple peptide bonds. So the polymer of this is called a polypeptide because I have multiple peptide bonds. Now, it's important to understand where that peptide bond is. There's been questions before asking you about that orientation, what's going to be paired together. So here we see a dipeptide, or is of course 2 amino acids bonded together. And I have this peptide bond kind of coded right here. You can kind of see it's right here between this carboxyl group of this amino acid and the amino group of this amino acid. So it's always between the carboxyl of the first amino acid and then the amino of the second amino acid. And I say first and second just meaning the one that's before the peptide bond and the one that's after that peptide bond, okay? So proteins have a directionality to them, okay? So there's an N terminus and a C terminus. The N terminus means it is on the amino side, and the C terminus is on the carboxyl side. So it just kind of gives you that long string. And proteins are built from their N terminus to their C terminus. So now, whew, that's so much information. We got to keep on adding more. You have a whole other half of the slide, right? So with this, we're going to see that our proteins are going to fold up. And so there's different levels to that folding, different levels to this protein structure. So the first level, or the primary structure, is just going to be the string of amino acids. Yes, this live stream will be saved as a video. It'll be posted onto Marco Learning's page, so you can access it later. So the primary structure is just a string of amino acids. And so between each of those amino acids, that bond is called a peptide bond, as we said before. And so, of course, we need to make sure that we know that that's the type of bond we have in primary. So you can see, of course, that primary structure. So the second level we have is the secondary structure. So secondary structure is an alpha helix, so it kind of makes like a coil. And then there's also the beta pleated sheet that just kind of like looks like a wave, okay? And that's why I make all these hand gestures to try to help you to understand, right? And so the bond that we see with this are hydrogen bonds. And they're hydrogen bonds between the backbone. So it's between a carboxyl group, and then it's between an amine group way downstream, like way far downstream. And so we're going to see that this kind of attachment between them allows you to get that coil for the alpha helix, or allows you to get that wave for the beta plate. And so again, it makes the structure for secondary alpha helix or beta pleated. And then the type of bond is a hydrogen bond, and it's between the backbone. The tertiary structure is going to be the third level that we'll see. And this is our final three-dimensional structure that we have, okay? Now, any bond that you've ever heard of is applicable now, right? We've got hydrogen bonds, we have covalent bonds, we have ionic bonds, you have van der Waals forces, you have all of these bonds taking place. It's important to understand what are those bonds taking place between. They're taking place between the R groups, okay? So that R group is going to bond with another R group, and that's what gives you that final three-dimensional shape, this final structure, right? Some of it has alpha helix, some of it has a beta plate, but it's all like configured together, okay? Now, tertiary is the end of a single polypeptide. So we're completely folded with this one polypeptide, but not all proteins are one polypeptide. So for example, hemoglobin. Hemoglobin has 4 subunits. So there's actually 4 polypeptides that will now interact in our quaternary structure. So this is the final level of structure of folding. And this has to do with, again, any bond, and is between the R groups of our different polypeptides. So now we can allow for these 4 subunits of hemoglobin to all interact to give us that structure of hemoglobin. So there you see our final little picture. Now I see a question coming up in the chat asking about R groups determining polarity. You are so right. So R groups, depending on them, right? If it's a hydrophilic group or a polar side chain, then it's going to fold toward the exterior or the outside of that protein. Why? Because the environment is aqueous and it's made up of water. And so since you have a hydrophilic or a water-loving side chain, it's going to aim outward towards that water molecule. If you have a hydrophobic side chain, it's going to kind of focus inward and go toward the interior of the protein to hide. Hydrophobic means that it's afraid of water, so it's going to kind of hide to the interior of that protein. And if it's charged, it'll go toward the outside of that membrane, right? It's not the outside of the membrane, the outside of that protein structure, the outside of that configuration. So you might be saying, well, how do I know if it's polar or if it's non-polar? So you're going to notice that the side chain, if it's charged, will have a plus or a minus. So that one's an easy giveaway. If it's hydrophilic, then you're going to see that it's going to be made up of, there'll be oxygens and sulfurs. There'll be a lot of electronegative atoms in it. Versus if we're looking at a hydrophobic side chain, it's going to be all carbons and hydrogens, because we know that carbon-hydrogen bonds are non-polar covalent bonds versus the oxygen-hydrogen bonds or the sulfur-hydrogen bonds are a polar covalent bond. Okay, so that gets us to the end of proteins. So let's move into nucleic acids. So our nucleic acids, what makes it up? Carbon, hydrogen, oxygen, nitrogen again, and then phosphorus, okay? So there's only 2 of your macromolecules that have nitrogen. That was the proteins, and that's the nucleic acids. And there's been questions before asking you to identify something that had nitrogen in it. So it's important that we know that the nucleic acids do have that nitrogen in it, so do the proteins. So we mentioned earlier about the Hershey and Chase experiment and how they were coding different things. So they used phosphorus to code our DNA, our nucleic material. So I'm sorry, our nucleic acid to determine the hereditary material. And they were able to see that that phosphorus was on the inside of the E. coli that they were testing. And so since the phosphorus moved from this virus to the E. coli, we were able to say, okay, well, that must be the hereditary material. And that is just like a quick overview of Hershey and Chase. We'll talk more about Hershey and Chase when we get into those experiments in unit 5, 6, later on. So what is a monomer? As we said always, you need to know those monomers. It's a nucleotide. And a nucleotide kind of has this general basic shape. So we're going to have a phosphate group, we're going to have a nitrogenous base, and we're going to have a pentose sugar. So these are going to be found in every single one of our nucleic acids. Now, these vary a lot based on whether we're in DNA or whether we're in RNA, and we'll talk about that in just a moment. So the bond that we'll see is a phosphodiester linkage, okay? So this is going to be between the phosphate of one, okay, and the hydroxyl of the other, because right here on this 3rd carbon is a hydroxyl group. You can see it right here that there's this hydroxyl, okay? And so we'll form that bond between the phosphate and that hydroxyl. So this right here is our phosphodiester linkage. Now, it's important to understand that DNA and RNA have a directionality, just like proteins. The directionality for this is going to be 5' to 3'. And when we look at this, it's going to be anti-parallel. So if you notice in our picture, we have a 5' end here, and across from it is a 3' end, showing you that they're anti-parallel. They're equidistant apart, but they're going in opposite directions. So that was why we called it anti-parallel. So here we see a long structure that we have. Okay, so again, our 5' end is going to have a phosphate group on it. So there's this phosphate right here, right? And then our 3' end is going to have the hydroxyl group. So my students long ago said we should just say poo, because it will help us to remember. So P for the 5' end and OH for the 3' end gives you poo. I don't know if that helps you, but that's what my students used to say back in the day. And so we have different nitrogenous bases. And so the different nitrogenous bases you can have would, of course, be adenine, thymine, cytosine, guanine, and uracil. 2 of those are going to be purines, okay? So what I mean by purine is that it is a double ring. So you can see here that there's 2 ring structures attached to each other. And then here with guanine, again, I have 2 ring structures attached. So those are going to be called purines. And so a way that you can remember that is you're looking at pure silver, because silver's elemental signal is Ag. So purines are Ag. And then the other ones we have are called pyrimidines. The pyrimidines are going to be the single ring. So here you can see with cytosine, we have 1 single ring. And then here with thymine, we have, again, a single ring. And uracil is going to look very similar to these with that single ring. So the way to remember it is, of course, cut, because you're going to cut the pyramid. I don't have a better way to remember it. Someone in a review session once told me that if you think about a pyramid being a triangle, there's 3 points to the pyramid. And so you can remember that CUT. I don't know. Well, it depends on how you want to remember it. In the base pairing, adenine pairs with thymine or uracil if we're looking at RNA, and then cytosine pairs with guanine. So it's important to know the number of hydrogen bonds that you're going to have here. So between adenine and thymine, we're going to have 2 hydrogen bonds, and between cytosine and guanine, we're going to have 3 hydrogen bonds. Okay. To remember that, it takes 2 lines in order to make a T for thymine. So there's 2 bonds, and then C is the 3rd letter of the alphabet. So you can remember there's 3 bonds between C and G. Hopefully that helps remember. And then we also need to know the differences between DNA and RNA. So we've already mentioned that they have a different in the nitrogenous bases, in which they have A, C, and G, but DNA will have a thymine or a T, while RNA will have uracil, so we'll see this U. The sugar is different. In DNA, we have a sugar of deoxyribose, but in RNA, we have a sugar of ribose. Somebody in the chat just says pyrimidines also have a Y, while A and C don't. So it's another way you can remember the pyrimidines. And then of course our strandedness. So double-stranded versus single-stranded. There does exist single-stranded DNA. There does exist double-stranded RNA, but we're looking at the general consensus of it, which is that DNA is double-stranded and RNA is single-stranded for the most part. And then when you talk about the sugar, I don't think I mentioned this, but when we say it's deoxy, students oftentimes think that that means that there's no oxygen in it. All that means is that they're missing one oxygen. So right here, you see that there's not a hydroxyl group, which tells you that it is missing that one oxygen. So if they give you a question in which you can see the numbers of carbon, hydrogens, and oxygens and whatnot, you can tell that it is DNA because it'll be missing one oxygen versus the RNA will have all of its oxygens. So that gets us to the end of nucleic acids. If you're wondering why I'm going so fast, I'm trying to get to the time for us to be able to do Q&A because the session's only an hour long. So I wanted to make sure that you had that time to ask your questions. So our fourth macromolecule we have is lipids. So now lipids are made up of carbon, hydrogen, oxygen, and then there's sometimes phosphorus. Phosphorus is going to be found in your phospholipids, but for the most part, you're only going to see carbon, hydrogen, oxygen. Now, when we saw the carbohydrates, it was a 1:2:1 ratio, but our lipids are going to be a 1:2 to few because there's not a lot there's not a lot of oxygen. It's usually just a couple oxygen molecules at the start of the fatty acid tails when they bind to the glycerol. So there's not a lot of oxygen. So 1:2 to few. Now, there is no monomer of your lipid because these aren't formed by a repeating structure. So when we looked at carbohydrates, we had a repeating structure of monosaccharides. When we looked at protein, we had a repeating structure of amino acids. When we looked at nucleic acids, we had a repeating structure of nucleotides. But lipids don't have that repeating structure, so there is no monomer for them. So instead, we have three different lipids that we talk about. I'm sorry, I missed this up. All of our lipids are going to be considered nonpolar because of the fact that they're made up of these hydrocarbons. So there's three of them we have. We have fats, we have phospholipids, and we have steroids. Our fats are going to be made up of this glycerol that you see right here. And then we have three fatty acids that come out from it. So you go through three different dehydration reactions in order to bond each of those fatty acids. Okay, so where you have three water molecules that come out of it. And so you can look at it and see that there's two that are having these straight change. And then there's one that's kind of like bent and it's turned. So you're thinking to yourself, well, why is it bent? Like what's happening there? So we have two different types of fatty acids. There are saturated fatty acids. What that means is that they are completely saturated. Every single carbon will have two hydrogens on it. It is completely full, completely saturated. There's all single bonds going through that whole fatty acid, all single bonds. Versus an unsaturated fatty acid is going to have at least one double bond. So if you look right here, you can see that we have a cis double bond. Cis meaning that they're on the same side, so these two hydrogens are on the same side of that double helix. Double bond. So we would call this a cis double bond. And so this is considered a unsaturated fatty acid because it has that double bond in there, which causes not all of our carbons to be saturated with hydrogen. Yeah, most of them are saturated, but we still have one that's not. So this is considered unsaturated. So if you've ever heard of like omega-3s, omega-6s, omega-9s, what that's telling you is that every third carbon has that double bond in it. So it's just kind of giving you information about the type of double bond. Okay, so that leads us to the end of fats. So phospholipids, all they did here is we take off one of those fatty acids, and instead we have a phosphate group attached. Okay, so you see we still have our two fatty acids coming down, and then we still have a glycerol. I used a cartoonistic picture just because it makes it a little easier to show like how this makes up the membrane. But there is a glycerol molecule right here, and that's attached to a phosphate group and then two fatty acids. So all we do is take off one fatty acid. Nothing too crazy here. And this is called amphiphasic. So we are specifically calling this molecule to be amphiphasic because it has a polar and a non-polar region. This one phospholipid is considered antipathic. But when you make an entire membrane of these phospholipids, we lose that antipathic component because that one little phosphate doesn't have enough polarity on it to form the whole membrane to be antipathic. So the membrane is still considered nonpolar. And so when it associates, it's going to associate so that those polar hydrophilic heads of phosphate go toward the extracellular and intracellular region of the cell, and that the fatty acid tails kind of go toward the interior because they have that hydrophobic interaction to kind of associate together because they're all nonpolar. Whew, that gets to the end of phospholipids. So steroids. Steroids are made up of 4 fused rings. You can kind of see 1, 2, 3, 4. And so you actually consume most of your steroid in terms of cholesterol. And then your body modifies that cholesterol by adding different functional groups to the cholesterol, which forms the different steroids that you're going to need. Well, steroids, we come back to a lot in terms of cell communication because steroids can be a type of hormone. You have steroid hormones, so like estrogen and testosterone. Those are steroid hormones. And so they are a part of cell communication. And so we'll talk later about this, but because of the fact that our steroids are nonpolar, we're going to see they'll have intracellular reception, which means that the receptor is going to be on inside of the cell because these can pass directly through that plasma membrane. And we'll talk about that a little bit more when we get into unit 2. Whew, so that gets us to the end of our macromolecules. You're thinking to yourself, oh my, Miss Jones. So I wanted to make it into like a kind of a graphic organizer for you. So this one page can be printed and you have all of that information that we had on all those slides. The only issue is right here, you don't see the picture of the protein structure. It's because of the way I had to format the structure, like the PowerPoint, I'm sorry. So let's get into our next part, which is water properties. So water properties all come down to the fact that water is polar, right? And a lot of the properties come from that. So like cohesiveness or the fact that it's universal solvent or the surface tension, all of those are due to the fact that water is polar. We also see that there's a high specific heat. It has less dense when it's a solid. And then also the pH of it is kind of important, kind of the connect in there. It may not actually be a unit 1 topic, but I felt like it went really well because we're currently talking about water. So let's jump down that polar rabbit hole, if you may. So as I said, universal solvent, cohesive, and then surface tension. So when we think about it, water has polar covalent bonds. So number 1, a covalent bond means that you have sharing of those valence electrons. So valence electron is electron on that outer ring. And so if 2 things are sharing that electron, it's called a covalent bond. Now, this is a polar covalent bond because oxygen is more or less negative than hydrogen. So it's going to pull it slightly, okay? And so because of the fact that the oxygen pulls that electron closer to it, it has more control of it, which means that it's partially negative, while the hydrogen has less control of it, and it's going to be considered partially positive. And so that bond between them is a polar covalent bond, which leads to partial positives and partial negatives within that water molecule. So because of that, we see a lot of hydrogen bonding taking place between them. So here you can see the hydrogen bond. So the partial positive hydrogen is attracted to the partial negative oxygen. I'm sorry if you hear a little bit background noise. My son is trying to get into my room. So we have this little hydrogen bond that's showing that attractive force. And so we see hydrogen bonds occurring between the oxygen and the hydrogen. That's going to be between our water molecules. And those hydrogen bonds are what we see being the reason why we have all these other things, right? So universal solvent, because of the fact that we have a partial positive here, I'm sorry, I went on the wrong one. Because we have this partial negative here on the oxygen, it causes anything that's partially positive, like polar, or anything that is positively charged, like a cation, to be attracted to that side. And the same applies for hydrogen. Because the hydrogen is partially positive, it attracts partially negative or anion things, okay? So we also have cohesion. Cohesion has to do with that water molecules are attracted to other water molecules, okay? And so that's cohesion. Versus adhesion is when water molecules are attracted to other polar substances. And so you kind of have this picture here, right? Where we have these water molecules are being attracted to each other, which shows you cohesion, versus adhesions where they're attached to some other polar or charged substance. You see this when we look at the meniscus, or that little U shape when you put water into a graduated cylinder. It kind of climbs the edges, right? And somebody in the chat is asking about capillary action. So cohesion plus adhesion together make that capillary action. It makes it so that the water is able to move from the roots to the leaves in a plant. How? Because up here, we're seeing transpiration, right? The water is evaporating. And as that water evaporates, it pulls a water molecule. And that water molecule is attached to other water molecules by cohesion. So we see like this pull of this string of waters up, right? And at the same time, the water is attracted to the sides of the xylem, or the sides of this tube that brings the water up. So it's going to kind of help to hold that water up so it doesn't fall due to gravity. So we kind of see cohesion and adhesion together. And I wouldn't call cohesion and adhesion one property together. There was a free response question a couple of years ago that separated them, and it was two different points. And so if you put them together, you would risk losing that point. So I probably would keep them separate properties. You could mention that it's all due to the fact that water is polar, but kind of you want to connect them, that's perfectly fine. So surface tension has to do with, again, cohesion. Oh, wait, there's no adhesion in this. That's the typo. So due to cohesion, all these water molecules are attached to one another. Okay. And so because of the fact that these water molecules are attracted to one another and attached to one another, they create the surface on the solution, right? And the solution tends to be, of course, water. So here you can see kind of gravity is kind of pulling that substance down, and you're able to kind of like hold that material up. You could do this with a paper clip. If you want to be like special, like, and pretend like you're magic, take a paper clip and a fork, and you can take the fork and dip the paper clip down into the water and then move the fork under, and the paper clip will stay on the surface. I mean, you can show people that you're magic. So this also allows you to skip rocks. So you can kind of throw a rock, and as long as you don't have too much force to break that surface tension, then it'll just skip along the top. Also, this is how a water strider moves across the top of the water. There's like a bug that just walks across. So that brings us to the end of the polar part of it. But let's talk about this high specific heat and then less density as a solid, right? So it's less dense as a solid. Why? Because the fact that those hydrogen bonds inhibit that compaction. It inhibits these water molecules from getting close together. And when they get close together, that's what ends up making solids more dense than their liquid component, right? So because there's this space, not air, but because there's this space in there, we're going to see that ice will float. So solid water floats. And this can act as a temperature buffer. So it can allow for like lakes and these different aquatic environments that kind of get really cold. It ensures that the entire lake doesn't freeze. It ensures that we can maintain a temperature that isn't too cold for these life forms in there, right? And so that they can survive the winter and be okay. So this picture kind of shows you what I was mentioning about how there's all this space inside that keeps them like apart, all this stuff. And it has to do with those hydrogen bonds. So we also have high specific heat. High specific heat just means that it takes a large amount of energy in order to change 1 gram of water by 1 degree Celsius. I mean, increase or decrease, it's 4.184 joules. If you really need to know the number, but it's okay. And so this allows us to have temperature control in coastal regions, right? So during the day in that coastal region, you've got the sun beating down on the ocean, right? And it's picking up all that heat and it's absorbing it. So you aren't feeling all of that heat on the beach, right? And then at night, when it cools down, that heat is released from the water, giving you a nice warm breeze. So it's kind of fun. This also works with body temperature. So if you're really, really hot, like you go out for a run, you go exercise, you're just hot, right? You start to sweat. So what's happening there? Well, that's evaporative cooling. It has to do with the fact that you have water on your surface and of your skin, and your skin is hot. So the heat from your skin is heating up that water. And as it heats up that water, it's breaking these bonds and it's converting the liquid water into gas water. And so we are able to evaporate, taking the heat with us. So this is called evaporative cooling. And then of course, leads us into pH. pH is calculated by having negative log of your hydronium ion or hydrogen ions. And so as the concentration of your hydronium ion increases, your pH will decrease. This comes into play when we start talking about cellular respiration, in which we're talking about like the hydrogen moving across the membrane. And so you're able to say, that the intermembrane region, intermembrane space is going to have a very low pH because of a high amount of hydronium ions in there due to the process of cellular respiration, right? Whew, there's a lot of stuff, right? So I have 2 multiple choice questions, and then I have a portion of a long story response question, because I figured it'd be good for us to just have this quick tip of checking information, right? So scientists examined a folded structure of a purified protein resuspended in water and found that amino acids with non-polar R groups were primarily buried in the middle of the protein, whereas amino acids with polar R groups were primarily on the surface of the protein. Which volume best explains the location of amino acids in the folded structure? So we have these polar R groups, and they're on the surface, which is true, right? And it says they can form ionic bonds. So ionic bonds, we know, are due to attraction between opposite ions. So an anion and a cation are going to bond together, which leads us into that ionic bond, right? Well, if we're looking at polar, then we're not looking at an ionic bond, we're looking at covalent bonds. So this actually forming some type of hydrogen bond between them if you're looking at that, okay? So we know that A is not right. B says polar R groups are too bulky to fit in the middle of the protein or pushed toward the protein surface. Again, polar is going to fold toward the outside of the membrane due to the polar nature of its aqueous environment. So we're not looking at that here. C says non-polar R groups that cannot form hydrogen bonds with the water are pushed into the middle of the protein. Well, that's true. If it's non-polar, then we're not going to have a partial positive or a partial negative, right? And we're not going to see that bonding with, or hydrogen bonding with, the aqueous environment on the outside, right? Plus, we're going to see all that hydrophobic interaction taking place, right? And then D says non-polar R groups from different parts of the protein form covalent bonds. No, it's not covalent bonds. It has to do with hydrophobic interaction, which is due to attractive forces. So the only logical answer we have was C. Okay, and this leads us into our next question, which definitely I feel like is not a Unit 1 topic, but it was coded as a Unit 1 topic, so I want to make sure that you realize that this can apply. A lot of the Unit 1 stuff actually is more in the later units because of the fact that it is the fundamental information, like the supporting information, to be able to then answer a lot of our biological questions. So Rosalind Franklin's X-ray diffraction images taken in the 1950s most directly support which of the following claims about DNA. So we already talked about all these structures of DNA, right? We know that DNA is anti-parallel, right? We know that it's made up of a purine bonded with a pyrimidine, right? Your purines was your adenine and your guanine. Your pyrimidines were your cytosine, your uracil, and your thymine. So you know that an adenine and a thymine bonded together is a purine and a pyrimidine. A guanine and a cytosine is also a purine and a pyrimidine. So they are going to have, of course, this equidistance. We know there's 2 strands that make it up. So there's all this stuff that we know about it, right? So now let's look at our answer choice and see which of those could be found thinking about a picture of X-ray crystallography. So the ratios of base pairs are constant. Can you see ratios from a picture? Well, no. The nucleotide sequence determines genetic information. Although that is a true statement that the nucleotide sequence can determine your genetic information, again, you can't see that from a picture, right? An X-ray crystallography picture. The 2 strands of DNA are anti-parallel. That is also a true statement. Can you see it in a picture? And the basic molecular structure is a helix. Well, that's true because of the fact that we can see that you've got an equal distance and it's making this like circular shape from that helix. So that was our answer there. And then our free response practice. So last year, 2021, they had one of the long FRQs. And I didn't want to bother you by showing you the entire long FRQ because only A applies to Unit 1. So the geneticist investigated the mode of inheritance of a rare disorder that alters the glucose metabolism, the first show symptoms in adulthood, all this fun stuff, right? So they say the disorder alters glucose metabolism, describe the atoms and types of bonds in the glucose molecule. So think to yourself for just a moment, see if you can come up with what the answer is, okay? There's a couple questions in the chat that I'm going to answer real fast. If you want to go ahead and type your answer to this and tell me what atoms and the types of bonds that are in a glucose molecule, see if you get it right. And I'm going to answer the questions that I currently see in the chat. So somebody asked, what is the difference between an ionic bond and a hydrogen bond? So an ionic bond is going to be between different ions, right? So a cation is a positive charged ion and an anion is a negatively charged ion. This ion is formed from the transferring of electrons. So one of those valence electrons was taken by another. So example would be sodium and chlorine. So sodium is positively charged because it lost its valence electron to chlorine. Chlorine is negatively charged because it gained that valence electron. This allowed both of them to have a complete octet. And now they, of course, have a charge because they have more protons than electrons, giving them a positive charge, or more electrons than protons, giving them that negative charge. Versus a hydrogen bond is due to a partial positive or partial negative. They have a covalent bond, so they don't actually lose their electron, they just have less control or more control of an electron. They're partially negative if they have more control of that electron, and they're partially positive if they have less control of that electron. So I'm giving you a moment to answer in the chat. So the different atoms that make up glucose, we know it's a carbohydrate. So it has carbon, hydrogen, oxygen, right? And then we know the type of bond that they have is a covalent bond. So you had to mention both carbohydrates, hydrogens, oxygens, and you had to mention that they were held together by that covalent bond. And that is the part one of this FRQ. So that gives us about 20 minutes that I can answer any questions that you have about Unit 1. My goal was to kind of do it this way, where you had like a quick little refresher of the concepts. And then you also had some time to ask questions. Back in the day, I used to do these sessions on Instagram, and they were 100% Q&A, in which you would ask the questions back and forth. You would ask me a question, and I would then answer your question. And so it allowed for more of a conversation and less of me just structured stuff. So I did want to give you a moment that if you had any questions, you could ask those questions. I'm currently seeing like the live, and so I see that you're a little seconds behind me. So the Q&A literally just popped up on that screen. So I know that you haven't even had a chance to ask questions yet. So I'm going to give you just a moment that you can now ask some questions. There were some questions about that FRQ. It says, thanks, the atoms in the glucose molecule, carbon, hydrogen, oxygen, 1:2 ratio, glycosidic linkage bond between the 1 to 4. You're correct. They do see a glycosidic linkage. I would assume that the readers would give you credit for glycosidic linkage because that is a specific covalent bond that we have. So I would honestly, actually, good point, Aiden. You're right. It wasn't talking about multiple electrons. It was only talking about the single one. So yeah, glycosidic wouldn't count. Because they were asking about a single glucose molecule. I'm sorry, I was forgetting the question. But yeah, you were right that between glucose molecules is glycosidic, but since the question specifically asked about one glucose molecule, it would be covalent. So I don't see any questions in the chat. Does anyone have questions, or do you want me to get John to end our session? Like, what y'all need? Hey, John.
Tiffany, I wanted to just chime in here. Great group of people, by the way. Thank you guys for watching. I wanted to also point out, this broader question. Today's February 6th. Some of you were watching this the night before the exam is recording, but what are some of the resources that you've got on your Instagram page, which is AP Bio Penguins, on your TikTok account, some of the things you're going to be doing this spring, because there's plenty more to review in the coming months.
Of course. So on my website, I have a ton of stuff. So I have a 316-page review guide that walks you through each of the standards, and it gives you ICANN questions. ICANN statements. There's topic questions in there. I've got multiple choice questions, free response questions, all broken down by the units. Actually, let me do it this way. I can show you the website. So here you can kind of see that I've got, a whole section here with all of that's where you would get the review guide. It's got all that. Every single day, I'm going live on my Instagram, not live on my Instagram, sorry. I post in my stories every single day questions about these topics. I will be going live every single Sunday, either on TikTok to do quiz games with y'all, or I'll be coming live with Marco Learning on the other Sunday so that we can go through all that. And all those sessions will be recorded and posted. If you go to the Marco Learning Sessions tab, you can also see, of course, all of the sessions that I've already done this year with Marco Learning. You can get all of the ones from last year. So if you click here, you can get to all of last year's sessions as well. You have all that. I have TikToks that I post, quick little 1-minute blurts about concepts. I've got 60 plus review games, and these are always open. Just go to this website, type this code in, and you can access these games at any point. If any of those games are hit their participant limit, there's actually backup games at the bottom, so you can still access them even if the codes are, ended. There's videos from FRQ Friday, so that if you are having trouble with FRQs, you can see, hey, this is how Ms. Jones would have approached it. This is how I can find points without actually knowing some information. Like, how can I pull information from the prompt? There's a lot here.
And Tiffany, this is what I so appreciate about everything you've done for the community of students and teachers who are in AP Biology, is that you've put out so much content for free. And I think that that's so generous of you and your time. I love that YouTube's a free platform where we can talk to you all. We are, did get a question from Victoria about our FRQ grading program. Now, because one of the things we did last year was one of our superpowers at Marco Learning was people would send us their essays, their free response questions, and we would grade them. But because we have people grading them, we have to pay them. That we can't make a free resource. But there's more that we're going to be posting about that on our Instagram page, on our TikTok account, on our website. So definitely stay in touch with us. And I think it's just an exciting time for those of you who are here this early to get ready. Take advantage of these incredible resources that Tiffany's putting out, that they're being built. And I can just say that we've got a great mascot at Marco Learning, but I love me a penguin too. And I think, Tiffany, if you could I've hoisted Marco up live on our YouTube channel. I think if you could just invest a few $10,000 into a little ice farm for a penguin, adopt it and maintain it with little fish for a year, it'd be really great to hold a live penguin up on air. At least we'll get some stuffed animal versions.
I'll work on getting a stuffed animal version for us, because at this point all my penguins are at school. I don't even think I have any penguins in my office. The best you can get is a mug that says I'm a penguin trapped in a human body. That's the best I can do for you. Sorry.
Put out some swag, some merch for all of this. Well, everyone, and Lila, you're saying that her content is so amazing. It is. Thank you so much, Tiffany. I don't see, actually, there's one more question that came through, and then I'll let you answer that one.
There's actually a couple questions I'm seeing. I see one about how are we supposed to know covalent bonds versus ionic bonds? So covalent bonds are due to sharing of valence electrons. So it's usually the materials that are kind of like carbon, hydrogen, oxygen, nitrogen, sulfur, kind of in that inner part of the periodic table, versus our ionic bonds are going to be, I believe, non metals and metal. So that's like chlorine, and that's sodium. Most of the bonding that we see is going to be covalent. And I believe that they're going to give you some type of information to be able to determine if it's ionic or covalent, because of the fact that technically knowing those bonds is not part of this, like the standard, right? You don't have to be able to identify them, but you do need to know what is which ones have covalent bonds in them. And all of our macromolecules are going to have covalent bonds in them. The only time you ever see ionic, you'll probably see it as a positive charge or a minus charge as an R group. And you'll just know that that has to fold, of course, outwards. So somebody asked, how does salinity affect the protein structure? It's going to denature it. Basically, anything you say has to affect the protein structure, most of the time it's going to denature it, or it's going to cause it to unravel. And it has to do with the way those bonds form. It will cause them to kind of disrupt those bonds. So if you change the pH, if you increase the temperature, all of that is going to cause denaturation. Decreasing temperature doesn't, because we don't have that kinetic energy that breaks them, but increasing temperature, changing pH, and salinity is all going to denature your protein. Let's see, somebody says, what molecules pass through phospholipids? molecules that can pass through phospholipids would be small and nonpolar because of the fact that it can fit between those phospholipids, as well as it has to be nonpolar to fit like to be able to pass through that nonpolar region. And I think that gets us to the end of the questions that I have in here. So with that, since I don't see that there's any other questions, John, you're welcome to, wait, somebody said something. Does it affect all four structures? It'll affect everything past the primary structure. So primary structure will stay intact, but the secondary, tertiary, and quaternary structures will all be broken down in some way, shape, or form. The end.
Great. Well, thank you so much, Tiffany. This was such a great session. Again, we're going to be going live on this channel, so make sure that you subscribe, turn on that notifications bell, and be in touch with us at Marco Learning and AP Bio Penguins. And you know what, Tiffany, we'll definitely have to set up some fun Instagram and TikTok lives on collabs on top of this, with everything we're going to be doing. So have a great day, everyone. And thank you, Tiffany.